Hello, everyone. We're going to go ahead and continue on in Unit 0 here. Today, in this video, we're going to be looking at topic 3 of these notes, which is standard error of the mean. In the last two videos, we talked about controlled experiments and what those look like and the different components of those experiments in science. We looked at how data is organized and displayed on different types of graphs. But today, in this video, things are going to get a little bit higher level. We're going to talk about what happens when we actually now have all this data and we're analyzing this data and trying to figure out, does this data actually support a claim that we're trying to make? Does it support our hypothesis? Does it support or not support what we were thinking was going to happen? And not just to ourselves, but to other people. Would my data actually be convincing enough or strong enough to other people to actually support what I'm saying? And so to do that, we have to use, in science, we use statistics to analyze our data to see if it actually is as strong as we might think it is, or to reveal to us if our data is not strong enough to even to make a claim. And so we're gonna look at that in this video. And there's two in this class. There's two types of statistical tests that we're gonna do on our data to see if our data is meaningful, or what we call significant data, or if it's not significant data. One of them is called standard error of the mean, which we're going to talk about in this video. And in the next video, we're going to talk about chi-squared, which is another type of statistical test that we can do with certain types of data to see if it's actually significant data to support certain claims. And so I want to start with an example to give you guys an idea of why this is important. So let's say I was there's two different types of fertilizers that I'm testing. There's fertilizer A and fertilizer B, and these two fertilizers claim that they help plants grow. And let's say that fertilizer A is campaigning and stating and claiming that they create or produce more plant growth than fertilizer B. And fertilizer B is saying, no, actually, our fertilizer creates more growth than fertilizer A. So let's say I set up an experiment, a super simple experiment. And I want to test that. I want to test which fertilizer is actually better for plant growth. And so let's say I have some plant seeds, and let's say it's a controlled experiment. It's the same type of plant seed that I'm going to try to grow in these two fertilizers. I give them the same amount of soil and fertilizer and the same size pot with the same amount of sunlight, the same amount of water, very controlled. And so, and let's just say I took one plant seed and grew it in fertilizer A and one plant seed and grew it in fertilizer B. So after just doing it once, let's say the plant in fertilizer A grew 5 centimeters and the plant in fertilizer B grew 8 centimeters. And so my first question to you guys is, does that convince you of anything? Does that convince you that B creates more plant growth than A? Do you think this data is strong enough to even be used as support to say that B might just be and looks like is better than A? Or is it not convincing enough? Is this difference not a big enough difference? Or the fact that I did it once wasn't convincing enough? Let's go ahead and step it up. Let's say I did this 10 times. So not just once, but I had 10 different seeds that I grew with fertilizer A, and I had 10 different seeds that I grew with fertilizer B. And so here's my data from fertilizer A from the 10 different plants, so including this plant with 5 centimeters and 9 other plants, and how much they grew after 2 months. And same thing with fertilizer B. And you guys can look at all these numbers, and you can look at all these numbers, and I don't know if that makes you think differently about anything. You might see some more smaller numbers over here, maybe some larger numbers over here. You might be thinking to yourself, it would be helpful if maybe we took the average of these groups. And so let's say I figured out the average. I added all of these up and divided by 10 to figure out the average. And I took the average of these, like what's the average growth after those 10 trials in fertilizer A and fertilizer B. And let's say here's the average. I already did it for you guys. The average for these 10 trials is 6 centimeters. For these 10 trials is 8 centimeters. So again, I would ask you, okay, what about now? Does this convince you that after I did it 10 times, on average, fertilizer B is causing more growth than fertilizer A? Do you think this data is strong enough to support that claim? Or again, are you not convinced? Are you looking at this and you're thinking, well, maybe if you did 10 more trials and another 100 trials, like how, maybe it's not the fact that B is better than A. Maybe these are just random differences that just happen to have occurred. Okay, it's just coincidental. And so that's the big problem in science, is when we get our data, how do we make sure that it actually is convincing enough or strong enough to actually be meaningful? And so that's when we use statistics. So what we could do is we could figure out the standard error of the mean for these two groups, which I'm going to talk about in this video. And that actually will reveal to me if this data is in fact statistically significant. If the difference between these two groups is statistically significantly different, because in that case, that would be convincing to other scientists if it's statistically significantly different. Or maybe I'll find that the difference between these is not statistically significant. And if it's not statistically significantly different, then that data is kind of that's not going to be strong enough data to say anything at that point, to say that one of these fertilizers is better than the other. And I actually went ahead and calculated the standard errors. You guys don't know what these numbers mean yet, but for this group it was 0.5 and this group it was 0.8. And that actually tells me, and you're going to learn this in this video, those two numbers, those standard errors, those values actually tell me that this data, the difference between these two fertilizers is not statistically significant, which means that this data does not support that fertilizer B is better than A. It also doesn't support that A is better than B. If anything, it supports that fertilizer A and B produce the same amount of growth in the plants. That's all I could really say. There doesn't seem to be a statistically significant difference between them. So that's what we're going to talk about. We're going to talk about standard error and how we use it and why I knew that by looking at these numbers. And so, like I already said, statistics are important in science so that we can actually analyze the data we get from our experiments and see how strongly that data supports or does not support our hypotheses. So the standard error of the mean, this is, or a lot of times we just say SEM. This is a common type of statistics we use in biology experiments, and in AP Bio, this is one that you guys need to be familiar with. You're going to see it all year long. And so basically what it's used to do, it's used to measure how much the data that you took from a sample group deviates from the actual population that you were trying to study. And so that might make more sense with an example. So let's say I want to figure out the average height of juniors at Buchanan High School. So I want to know I want to make a statement about what the average height of juniors is at Buchanan High School. So instead, well, I could hunt down every junior at Buchanan High School. There's close to 700 of them, and measure all their heights and find the real average height of juniors at Buchanan High School. And that would be a lot of work and very annoying. And in science, a lot of times we don't want to go find every single lion in the savannah or every single tree in the rainforest. And so what we do is we find samples and we use sample sizes to get a good idea of what might be true about the real population we're studying. And so instead, let's say out of convenience, I just want to measure the average height of juniors in my classes. And so in my AP Bio classes, I have a good amount of juniors, and so maybe 50 or more juniors. And I'm just going to take the average height of those juniors, and I'm going to trust a little bit that that average is close, hopefully, to the real average height of juniors at Buchanan High School, the actual population. What I would do then, if I did take a sample, like let's say 50 juniors, not all 700 of them, and I measured their average height, I could then calculate the standard error for that average, for that mean, the standard error of that mean, and that standard error will tell me how closely that sample probably matches the real answer, if that makes sense. So basically, you could end up with a larger standard error value or a smaller standard error value. Don't worry yet about what's considered a high standard error or a low standard error. It all depends. But let's just say, in general, when you have a higher standard error, when that value for standard error is higher, that means two things. It means, first of all, the data that you took from your sample probably doesn't accurately represent the actual population. So if you have a large standard error, it means that very likely the average that you got from your sample it's very likely that it might be kind of far off from the real average you were trying to find. That's what a large standard error means. It also means a second thing. Having a large standard error also means that there was probably a lot of variation in the data that you collected from your sample. So when I was measuring the height of my juniors, and I was measuring how tall they were, and so maybe if I had a large standard error for that group, that would mean there's probably a lot of variation in that group. Maybe I was measuring people and they were like 5'7", 5'8", 6'3", 4'11", 4'5", 5'3". There was a huge variation in the data, a lot of different data points in that group. Now, if you have a lower standard error, what that generally means, that means that the average you got from your sample probably is close to the real average for the population you were trying to understand or figure out about information about. So it means that your sample probably is accurately representing the real population, and that your average is probably pretty close to the real average. And it also means a second thing. It means that there is probably little variation in your sample group. And so if I was measuring the height of juniors and I had a low standard error, what that would mean is that the data points that I collected probably didn't vary a whole lot. So maybe some people were 5'5", 5'8", 5'7", 5'9", 6 foot, 5'9", 5'7", 5'6.5". Like all the data is a lot more closer together. That's what a lower standard error means. So that's something you need to memorize about standard error so far. Just know if you have a larger standard error, it means that your average is probably not very close to the real average. I mean, it could be, but probably not. And if you have a small standard error, it means that your average is probably closer to the real average. And then another thing that it means is if you have a large standard error, there's probably a lot of variation. If you have a smaller standard error, there's less variation. So those are things you just need to know about standard error, even though we haven't fully understood it yet, so don't worry. There is formulas that you're provided that have to do with standard error on your AP Biology formula sheet. So when you guys take your AP test, and in this class even, you guys will always be provided a formula sheet. And this is just a snippet of some of that formula sheet. And on that formula sheet, it has an equation for finding the mean, and then what's called the standard deviation, and then ultimately the standard error of the mean. Now, here's the good news. You are not responsible for calculating ever the standard error of the mean, which is based off the standard deviation, which you're also not responsible for calculating. So I will never ask you in this class, and no one on the AP test will ask you calculate the standard error of the mean, because it's a lot of work to do by hand, and usually we just use a computer or a calculator to do it for us. Now the mean, you guys should know how to calculate the mean, which is the average. If I gave you a bunch of numbers and I said, find the average of those numbers, you should be able to find the average. Now this formula is not too helpful because it looks weird, but basically this formula is telling you, take all the numbers, add them up, and then divide by the number of values that you had. So I'm sure you guys have done that since elementary school. That you do need to be able to do. But calculating the standard error, which to calculate the standard error, you need to know standard deviation. Calculating those two things you don't need to do. So on a test question or on any type of assignment or question you have in this class, I will give you the standard error if you need it. I will calculate it for you, and that value will be given to you, kind of like what I did in this example. Like that value is just there. Someone already calculated it. Now what you do with that information, that's going to be on you. So now you're going to have to make meaning of what to do with that value and what that value ultimately means for your data. And so let's talk about that. So kind of what I was suggesting at the beginning of this video, data in science only supports a hypothesis if it's statistically significant. So if you want to make a claim and you have data that you think supports that claim, well, just because you think it supports your claim and you think, look, this data is convincing enough, that plant grew more than this other plant, I think that proves that I'm right. That supports what I'm trying to say. Well, no one cares what you think. Like no one in science, no other scientist is going to care if you personally feel like your data is strong enough. And so again, we have to use statistics on that data to show that it's statistically significant. Then people will care. Okay, then it's actually something that you can claim that's supporting what you're saying. And so in order to determine if two groups are significantly different from one another, not just different from one another, but significantly different, or statistically significantly different from one another, the averages of those two groups with plus or minus 2 standard errors should not overlap. And we're going to dive into that statement right now in a second. But this is going to be easiest to figure out if you graph the means with the error bars. And so you might have seen graphs like this before, like a bar graph or even like a line graph. And on these data points, on these bars, for example, there's these lines you see on here. I'm sure you've seen probably something like this on a graph before. You might not have known what these lines are. These lines are called error bars. And so they're used to show the average with plus or minus the standard error. And so, which is going to help you figure out if the difference in these averages between these groups are actually meaningful or not. I'll explain in a second. But these are called error bars. And to create the error bar, what you do is you take the average. So the bar represents the average. So let's say on this graph, they're looking at the effectiveness of different insect spray. And so insect spray A, it looks like there was 15 insects using that spray around. That's the average for spray A. Maybe they tested lots of, they did lots of trial of spray A, but the average was around 15. Now this error bar, what they did is they took the standard error for this group and they added it twice and subtracted it twice from the average to get this error bar. So the error bar, in this class at least, you will always create the error bar by doing plus 2 standard errors and minus 2 standard errors, which again, I promise this will make a lot more sense when we do an example in a moment. But that's what this graph is doing or showing you. It's showing you the average with plus or minus 2 standard errors. That's what this error bar is. Now, when you create the error bar using plus or minus 2 standard errors, this is going to give you 95% certainty that the differences between the two groups is actually a real difference, a statistically significant difference, and not just due to random chance. And that's going to be true if the error bars do not overlap. So look maybe over here real quick at this graph. Let's just look at the top graph. And if you look at, let's say just spray A and B, and if you look at the average between spray A and spray B, it looks like spray B has more insects after using spray B than spray A. On average, right? But to figure out if this difference is actually a real difference, because it could just be by random chance, by the random samples that you had using spray B and the random sample that you had using spray A, and you took the averages, it could just be by random chance B ended up higher than A or A ended up lower than B, and it has nothing to do with the effectiveness of these sprays. But it could also be because spray B just causes there to be more insects than spray A. It could be a real difference. Now, to figure out if it is a statistically significant difference between these two groups, A and B, you need to look at the error bars and see if they overlap with one another. And so it's a lot of times teachers will suggest students or imagine you had like a ruler that was flat, left to right, and you were sliding that ruler down across this, and if this error bar at any point overlaps with this error bar, then that means that those two groups are not statistically significantly different. So like in this example, I know that the difference between spray A and B, it's not a statistically significant difference because the top of this error bar overlaps with the bottom of this error bar. Like there's overlap between them in this region where my mouse is, they're overlapping. And so when that happens on your graph, that means the difference between those two groups, it's not real. It's real, but it's not meaningful. It could just be because of random chance. It's not strong enough to mean anything. Ultimately, it really means like spray A and spray B have the same effect. Like in this difference that you see, it's not real. Like it's not meaningful, so you're just going to pretend there is no difference. Like they have the same effect. Now, if they don't overlap, like look at spray C and D. And if you look at the averages, you can see that spray D had more insects than spray C, right? But if we want to know if this difference is real, a statistically significant difference, you have to look at the error bars. And if the top of this error bar does not overlap with the bottom of this error bar, which you can see in this example, the top of this error bar does not overlap with the bottom. There is no overlap between these two groups and their error bars. That means that this is a statistically significant difference between spray C and spray D. We would say then here, this difference does support the claim that spray D leads to more insects than spray C. And this data supports that claim because this is a meaningful difference. It's probably not due to random chance. And when they don't overlap, it actually gives me 95% certainty that there is actually a real difference between them. So that's what I was saying earlier. When you create these error bars using plus or minus 2 standard errors, and they end up not overlapping between two groups, that means that there's only less than a 5% chance that this is due to random chance, this difference we see between these two groups. And there's a 95% chance that this is actually a true difference, that there is actually something going on here causing D to have a different outcome than C. That's what that means. And in science, most of the time, a lot of the times, especially in biology, and a lot of published research papers, 95% certainty is the threshold you need to be accepted by the scientific community for other people to think or to feel that the data supports what you're trying to say. And so that's what you're looking at. So like if you look over here, you can even use it on a line graph. So this is showing you how the plant mass is changing over time, so how much the plant rate weighs, the mass of the plant over time, week 1, week 2, week 3, week 4. You can see the mass is going down after between week 1 and week 2, and the difference is statistically significant. So this is not on average, the plants, their mass is decreasing, and it actually, since the error bars, the bottom of this error bar and the top of this error bar do not overlap, this is a statistically significant decrease. Like I could claim that in between week 1 and week 2, there's going to be a decline in the plant mass. This data supports that. Now between week 2 and 3, you can see there's a decline, but the error bars are overlapping, which means that I can't say that in between week 2 and 3, there's going to continue to be a decline. This data is not strong enough to support that because the difference between these two data points here, this average and this average, is not statistically significant because the error bars overlap. And when the error bars overlap, it basically means that there's no difference between them. Even though you see a difference, that difference isn't real. It's not strong enough to actually be real or mean something. If anything, it just means it's the same outcome. So hopefully that makes a little bit sense visually with what's going on with these error bars. In your notes, you're going to have some questions to answer based off of this data table and this graph. On the left side of your notes, there's some questions about this. On here, it's showing you that in soil type A, the average growth, so here's different soil types, and here's after 1 year how much the plants grew on average. Again, each of these groups represent an average, which means there's probably lots of trials done. For soil type A, they tested 10 plants or 20 plants or 1,000 plants, but this was the average. And this is the error bar for that group. And the way they got this error bar, so you can see the average was 20, so that's why the bar says 20 on average. That was the plant growth for this soil type. Now the error bar, the standard error for this group was 2. So for all those plants that were given soil type A, the standard error was 2. Someone calculated that. You don't have to worry about it, but it's 2. And so what they did to create this error bar on this graph is they doubled this and they added it and subtracted it from 20. So they're going to do 20 plus 2 standard errors and then minus 2 standard errors. Now the standard error, one standard error is 2. So you're going to double it because you have to do plus or minus 2 standard errors. So instead of doing plus or minus 2, you're going to do plus or minus 4. You're going to add 4 to 20 and minus 4 to 20 because you have to double the standard error. Okay, so instead of for at 20, you're going to add 20 to 24. That's the top of your error bar, which you can see that's about 24. And then you're going to minus 4 from 20, which is about 16. And you can see that here. That's the top and bottom of your error bar. That's what they did to each of these. Like in this one, the average was 30, but then they're going to add and subtract 5. So you're going to add 5 and minus 5 from 30 to create this error bar. And that's how they set up this graph. And then once you can see that visually, you can start to look at these different groups and see which groups have statistically significant differences between them. and in which groups there's not statistically significant differences. Even though the bars might look different, you have to be careful. Just because the bars, the averages look different, that doesn't mean there's actually a real difference. That's just the average from a sample. And that doesn't mean that really there is a real difference. You have to look at the error bars to see if they are overlapping or not. And so that's why I know between soil type B and C, they're basically causing the same amount of plant growth. According to this data, B does not cause more plant growth than C. A lot of students might think that because the bar is higher. They're like, well, Mr. Austin, B, the average growth was 30 and C was 27. What do you mean it didn't cause more plant growth? Well, if you believe that, that's fine, but this data doesn't support that. This data is telling you that this difference right here, it's not statistically significant, which means we can't claim, this data isn't strong enough to claim that B was better than C. If anything, it's showing us actually that B and C had the same effect on these plants on average. In reality, not just in samples, but in all plants, the difference is the same. That's what this supports. The plant growth is going to be the same between them, even though from the samples, the averages were different. So just kind of start accepting that averages, when you look at samples, the averages could be different, but just because the averages from samples are different does not mean that in real life, outside of those samples, that there's actually meaningful differences between those two groups that you were studying. And you can only say there is a difference if there is a statistically significant difference, okay? So at the end of this, I want to go ahead and practice one example with you guys. At the end of, so you guys have these questions on the left side of your notes, and then at the very end of topic 3, if you look, I think it's on the next page, you'll see these last 2 examples. I want to do this very last one with you guys in this video. I'll leave the first graphing one to you, but we're going to create a graph together. And I just want to make sure you guys understand and feel comfortable doing and creating a graph using this data, and then looking at that graph to start seeing where there is statistically significant differences between different groups that we were testing. And so let's do this example together. Find this one in your notes. It's at the very end of topic 2 before topic 4. It says the goldfish experiment from topic 1 was performed by a researcher who repeated the procedures with 8 different fish for each group tested, meaning they performed 8 trials for each change in water temperature. So let's continue on with this fake example I came up with in topic 1 with the goldfish, and we changed their water temperature, and we're looking at how their respiration rate is affected by that. But what they're saying here is that they're going to do that 8 times for each group. So now we're looking at averages. So this is, they did 8 different fish and made the water 10 degrees colder. And for those 8 fish, this was the average change in respiration. And they took 8 other fish, and for those 8 fish, they did this experiment with those 8 fish and made the water 5 degrees colder, and that was the average change in respiration rate. Same thing with 0, this was the average. And when the water went up 5 degrees, this was the average. And so here are averages now from testing not just 1 fish per group, but 8 fishes per group. And then the standard errors, there's standard errors here already calculated for you for each group. Each group is going to have its own standard error value because it had its own unique set of data for those 8 fish and those 8 fish. So there's the standard error values. So what it says, it says using the template provided, construct an appropriately labeled line graph. Between which changes in water temperature is there a statistically significant difference in the fish's respiration rate? So let's construct this graph together. I'm going to pull up my camera here, and I think I have this printed right here. Actually, let's look at this sheet right here. Sorry, I'll focus it. A little better. Okay, so I want to take this data and I want to graph it here. And so we actually already made a graph in the last video using some basic data, but now we're looking at averages, and it specifically asks us to make a line graph. So we're going to make a line graph, which is kind of almost like the one we made. We made a scatter plot, but this time we're going to connect the dots, and I'll show you what that looks like right now. So we're going to go ahead and again do the independent variable here, which is the change in water temperature. We have a set of continuous values that exist on a continuous range. So we're going to go ahead and scale this. I have a 20 by 20 grid for you guys. This is 20 boxes by 20 boxes this time. And so if I have 20 boxes here and I have a range of -10 to +10, I can actually fit all of that with every unit equaling 1, which is the best scale. If you can make the scale 1 unit equals 1, that's awesome, because then it's really easy on your brain. So I could start at -10 and make each of these 1 and get to +10. So I'm going to go ahead and do that. This is going to be -10. And then this would be -9, -8, -7, -6, -5. I'll go ahead and label that. -4, 3, 2, 1. This is going to be 0 right here in the middle. And then we're going to have 1, 2, 3, 4, 5. And then 6, 7, 8, 9, 10. Okay, went ahead and scaled that. Now, and then let's label it. That is the change in water temperature. And then on the y-axis, we're going to go ahead and plot the dependent variable, which is the average change in breaths. Now, these values are a little bit different than the values I gave you before, because this is 8 fish in each group. This is the average, which changed these numbers a little bit, but it goes from -16 to +17. Now, here's the thing. You're going to pause before you start scaling this. Not only are we going to plot these averages on our graph, we're also going to have to create error bars. And for the error bars, those error bars are going to go ±2 standard errors for each of these averages. And so you need to make sure that your graph is not only going to fit these averages, but also the error bars that are going to go +2 and -2 standard errors. And so I'm going to go ahead and brainstorm my error bars real quick, which I strongly suggest you guys do that throughout the school year, is that you kind of think through all the numbers first before you start setting up your graph, because you don't want to set up your graph and then realize something's not going to fit, like an error bar is going to have to go up here, and you didn't leave room for that, and that's not allowed, so then you would have to restart your graph, which would suck. So for example, if the average is -16 for this first group, and the standard error was 2, well, we're going to double that, so that's going to be 4. We're going to add 4 and -4 from this. So if I minus 4 from it, that's going to be -20. And then if I add 4 to it, -16 plus 4, that's going to be -12. So what this tells me, I'm just jotting this down, the error bar is going to go from -20 to -12 on here. And so I need to make sure that my graph is going to fit those values as well, not just -16, but it can go from -20 to -12. That doesn't make sense yet. You'll see in a moment. But I'm going to go ahead and do that for each of these. So -11, I'm going to add and subtract 2 standard errors. The standard error is 2.5, so I'm going to add and subtract 5. So -11 minus 5 is -16. And then -11 plus 5 is -6. And then for this group, the average was -2, but I'm going to add and subtract 8. Don't forget to double it. So I'm going to add 8 and subtract 8. If I do -2 minus 8, that's -10. And then if I add 8, that's going to be +6. And then 14, I'm going to add and subtract 4. So if I subtract 4, that's 10. If I add 4, that's 18. And then for 17, I'm going to add and subtract 3. If I minus 3, that's 14. If I add 3, that's 20. So you can actually see that my largest and smallest value is not -16 and 17, but also the error bar, -20 is actually the lowest point that needs to fit on here, and +20 is the highest point that needs to fit on here, which is ultimately a range of 40 from -20 to +20, which I have 20 boxes right here, so I could just double that. I can make every unit equal 2 for my scale, and that will actually perfectly fit my data. How convenient. I wonder who came up with these numbers. So I'm going to start at -20. And I'm going to say every unit equals 2. I'm not going to label each one, but this is -20, -18, -16, -14, -12. This would be -10 right here. -8, -6, -4, -2. This will be 0. And then hopefully I'm counting correctly. This is going to be 2, 4, 6, 8, 10. And then 12, 14, 16, 18, 20. There we go. So I have a consistent scale. Every unit equals 2, and I'm going to fit my lowest value, my highest value on here, thankfully. And I'm going to label this. This is the change in respiration rate. I'm going to put change in RR. And the unit, don't forget the unit, this is breaths per minute in my fish. So let's start plotting. Here's what this is going to look like. Just like last time, we're going to plot dots for each of these data points. So the average for when the water went down 10 degrees, the average change in respiration was -16. So that's going to be right here. Be very careful with your negatives. That's -16. Okay, and then let's go ahead and do that and the error bar at the same time. So we're going to make the error bar. Even for a line graph, you can also make error bars for the data points. It doesn't have to be a bar. So in the notes, you saw an example where it was a bar and you did plus or minus to create the error bars. And so we're going to do that with the dot. So my error bar, I already did all the work. It's going to go from -20 to -12. So -20 is down here. So I'm going to make a line right here to -12, which is right here. And then I'm going to draw a line like this. So that's my first data point with an error bar. You can see the error bar symmetrical. That means I added everything correctly and hopefully plotted it correctly. So there's my first point. Now for -5, the average was -11. I'm going to plot that. That's right here. And then I'm going to make the error bar. The error bar is going from -16 to -6. So that would be right here, -16 to +6, to -6, which would be, again, be careful with your negatives. It gets tricky, your head gets tricked. That would be the error bar, which doesn't look right. And that's because this was not -11, that was -12. See, messed up. Negatives confuse me. So my dot should have been at -11, which is actually right here, between -10 and -12. That looks better. And then my error bar, again, is -6 to -16. That looks good. And then for 0, the average was -2, which would be right here. And then I'm going to, the error bar, that one's a big error bar. It goes from -10 to +6. So -10 is right here, make a line. And then +6 is right here, make a line. There's the error bar for that. Hopefully that's right. And then my next one for 5 is 14. I'm gonna focus here. Let's see, 5 and then 14. This is 10, 12, 14. And that one goes from 10 to 18. So 18 would be right here, and 10 would be right here. And then the last one is 17. So that's going to be between 16 and 18 right here. And that goes between 14 and 20. So this is 20 right here, and then 14 right here. Okay, and then I've plotted those points. And then at this point, I'm going to go ahead and connect them with a line, because they wanted a line graph, which I could represent this as a line graph. And so I'll even use a straight edge. So I'm going to connect these dots and these dots, these dots, and these dots. So you can see a line graph shows me that it's how it's changing at different values, but it shows me that there's different things going on at different values. Like it's changing a lot between these values and not so much at these higher values. But anyway, that would be the graph for this. You could also come up with a title. You could use the same title as last time when we used it in topic 2. I think I used fish response and respiration rate to changes in water temperature. I'm too lazy to write that right now. But now you're going to answer this question here. It says, between which changes in water temperature is there a statistically significant difference? So if you look here, when the water changes temperature, sometimes there is a significant difference between them and sometimes there's not. And so for example, between when the water did not change temperature versus when it changed -5, now that I have an average with the standard error values and I created these error bars, It does show that the average for those fish that I tested, the average went down, but it's actually not a statistically significant difference in respiration rate, in the change in respiration rate. So because these error bars overlap, this data actually is not strong enough to say that when fish are put in -5-degree water, or water that's 5 degrees colder versus water that's the same temperature, this data doesn't support that their respiration rate goes down, okay? Which is mind-blowing to some students. They're like, what do you mean? Like, look, it does go down. The respiration rate is decreasing versus when the water doesn't change temperature. But this data isn't strong enough to say that. If anything, it shows that when you put fish in water that's the same temperature versus when you put fish in water that's 5 degrees colder, their change in respiration rate is the same. That's actually what this data does support, because there's not a statistically significant difference between them. Now, if I was looking at -10 versus 0, now the error bars do not overlap. If you look very carefully here, the top of this error bar does not overlap with the bottom of this error bar. There's no overlap. There's a small space between them. And so that's a statistically significant difference. So then I could say when fish are put in water that's 10 degrees colder, their respiration rate does decrease more than when the fish are put in water that's the same temperature, or when the temperature of the water doesn't change for the fish, because that is lower. And not only is it lower, but it's statistically significant, okay, their change in respiration rate. And you can see on the other side over here, you can see some differences, again, that aren't statistically significantly different, but there are definitely some that are statistically significantly different. When the water gets even 5 degrees warmer, that does cause a statistically significant difference in the change in respiration rate. And versus when the water doesn't change and 10 degrees warmer, it's also statistically significant between this group of fish and that group of fish. But between these two groups, 5 degrees warmer and 10 degrees warmer, there's really no difference in how it affected their respiration rate because the error bars do overlap. So you guys would answer or point out some of those observations and describe them over here in this space. But I hope that is starting to make sense. We're going to be looking at that throughout the entire year. So hopefully it'll start clicking more and more the more we do and look at examples. But that's basically it for this video. Thank you guys. I will see you later.